Thank you so much for tuning in to this first video on Chapter 1. The goal here is to give you my explanation of what the authors are trying to convey to the reader. In my opinion, Chapter 1 can be neatly divided into 5 modules, each discussing important concepts. Module 1 talks about life, a definition of life, what it means to be alive, and what discriminates life from non-life. These 3 words from the word bank are important for the students to relate to. In the upcoming slides shortly, I'll be talking about the definition of life and how all living things are made of cells, and within those cells there's DNA, and the cells are surrounded by a plasma membrane. Your task is to answer these 2 questions in depth by the conclusion of this module. The first learning outcome is for you to define life. The second learning outcome is for you to list the 3 tenets of the cell theory. The cell theory is a very important concept in biology. One of the challenges to defining life is to come up with a statement which encompasses all living things alive today and in the past. Represented in this figure is a small selection of cell shapes found in nature. Whereas cell shapes may be unlimited, the size of cells is limited by various physical and chemical factors to the range of micrometers. At this time, students should have a very good sense of what a micrometer is. One of the major goals in biology is to come up with a single definition in which all living things fit. Unfortunately, we have not reached that consensus yet. So there is no single definition which applies to cover all living things. So where are we? At this point in time, we have a list or states of properties which all living things exhibit. For this class, the following acronym, Mr. Gray Plus, suffices to help us remember all the components that living things share. The M in Mr. Gray tells us that all living things metabolize. The R tells us that all living things respond. The G shows us that all living things grow, either in size or in number. The R, the second R, is to convey the meaning of reproduction. All living things have to reproduce. And finally, the E in Mr. Gray is to tell us that evolution is a very important component of biology. Therefore, all living things, in terms of populations, evolve. We have to add the plus sign to Mr. Gray to convey the meaning that all living things are made of cells. A cell is the fundamental unit of life. This is very important. Adding a little information to the previous slide, the word metabolize encompasses all chemical reactions taking place inside living organisms. These involve anabolic reactions, those that build up simple chemistry into complex chemistry, and catabolic chemical reactions, which do the reverse. Students tend to forget the response component of Mr. Gray. This simply says that all living things, as long as they are alive, have the ability to respond to their environment, both in a good sense and in a negative sense. The plus sign needs further qualification too. All living things are made of cells, but those cells have 2 important components. The first is DNA, and the second thing is all cells are surrounded, at least by one plasma membrane. Our textbook has a couple of other components that could be added to Mr. Gray to make it more complete. Now that we have a reasonable understanding of what entails life, let's move on to the very first theory in this textbook, the cell theory. The cell theory was first proposed in 1839, almost 200 years ago, and it has 3 important components. The first is that all living things consist of 1 or more cells. So the smallest living thing on the planet should be 1 cell in size. The second component of the cell theory, the second tenet as we call it, is that the cell is the basic unit of structure of all living organisms. So you cannot get any smaller than a cell and still be a building block of living creatures. The third tenet of the cell theory is that all living cells in existence today have arisen from pre-existing cells. So paraphrasing the three tenets into a simple list, we see here that all living things are made of cells. A cell is the smallest thing that is living. And number three, all cells come from existing cells. The second tenet is the one that students tend to forget most. So here is a typical low-level question that you could be asked on an exam or a quiz in the future, either in this class or other classes. And the question then is, are viruses, all of them, living? And the answer will be basically to go down the list of Mr. Gray plus and see if all items are checked. If they are, then viruses would be living. But if one of the items is not checked, then viruses would no longer be classified as living entities. And as we now know, viruses are not made of cells, so therefore they fail that test. As we reach the end of this module, the student learning outcomes are displayed once again to make sure that the student can now answer these outcomes. So basically, are you in a position to define life? And are you also in a position to state the three tenets of the cell theory? Should you have any problems with either one of those outcomes, this is a good place to stop and to go and reread the chapter, study the PowerPoint, and to listen to this video again. The next module is laid out in very much the same manner as module number 1. Module number 2 deals with the classification of cells into discrete types. These 7 words from the word bank are relevant for this particular module. Archaeon, bacteria, eukaryote, evolution, nucleus, prokaryote, and protozoa. Many biology textbooks, when comparing living organisms, use the terms unity and diversity. The unity is basically describing the features which are shared by all living things. The diversity component is describing those features which distinguish living things from each other. Learning outcome number 3 wants you to define the number of domains into which modern living things are classified. And then finally, the last learning outcome wishes you to compare and contrast eukaryotic versus prokaryotic cells. Let's look at the unity component of life. This basically says that these features are shared by all living things. This list is quite expansive and it begins at the basic level. All living things are composed of cells. All living creatures follow Mr. Gray plus. All living things contain DNA as a molecule of inheritance. So basically we've covered this in the previous module, but there's more. There's also this central dogma concept. The central dogma concept basically says that biochemical information flows from DNA through RNA to protein. What we have never found is information encoded in DNA being transferred directly to proteins in living systems. Another feature shared by all living cells is the process of cell division, whereby one mother cell divides into 2 daughter cells. Regardless of which organism we study, cell division is a core process which cells use to reproduce. There are many more features which all cells share in common, and those we'll cover in subsequent chapters of this textbook. Moving on to differences between cells, we enter the diversity of life section. Now, at the most fundamental level, there are two types of cells on this planet. Those that contain a nucleus, which we call eukaryotic, and those that lack a nucleus, called prokaryotic. Indeed, the word karyotic refers to the term nucleus in the Greek literature. So pro means before the nucleus, and eu means true nucleus. So if you discover a new cell under a rock, the only thing you have to do is to look for a nucleus. If you find a nucleus, it must be a eukaryotic cell. If it lacks a nucleus, it must be a prokaryotic cell. There is no other option. The next level of classification of cells is into domains. So prokaryotic and eukaryotic cells can be broken down into three major domains. Basically, the prokaryotic cells are subdivided into two domains. We have the eubacteria, which means true bacteria, and then we have archaea. The eukaryotes stay as their own domain called eukaryota. In summary, the three domains of life are eubacteria, number one, number two, archaea, and number three, eukaryotic. If one was to move on to the next lower level of classification, then the eukaryotes would be subdivided into four kingdoms: the animals, the plants, the fungi, and the protists. When differences between the kingdoms of life are input into a computer, the program rearranges organisms based on how similar or how different they are. And the map which emerges looks like this one presented in this figure. As one can see, all the bacteria species sampled are grouped together into this branch. The archaea are branched together here in the center, and all the eukaryotes are branched together on the far side. And as you can see, there's a relationship between the three domains of life. And what this basically says is that the archaea are very similar to the eukaryotes overall when compared to bacteria. So it's wrong for students to assume that bacteria and archaea are cousins of each other, when in reality it appears that they are not. A medium order assessment question could combine Module 1 with Module 2 to come up with a question such as this. How would you determine if something you discovered was living and then classify it? Let's take a deeper look at prokaryotic cells. Just as a reminder, the spelling for prokaryotic can be with a C or a K, depending on which textbook is being utilized. So the prokaryotic cell, the prototypical prokaryotic cell, are very diverse. They come in various shapes and sizes, and they are the most diversely dispersed life forms on the planet. They've been found 5,000 feet below the frozen Arctic ice in underground lakes. They've also been found 2 miles below the seabed when people have been drilling. They've also been discovered on top of Mount Everest at 29,000 feet above sea level. And believe it or not, they've also been discovered growing on fresh lava rock, which has barely cooled down. So to say that these creatures are resilient is an understatement. The world of prokaryotics, as we discovered earlier, is divided into two of the three domains of life: the eubacteria and the archaea. As we mentioned in the map that we showed you earlier, there are major differences between the eubacteria and archaea. In general, you are familiar with eubacteria, E. coli being a great example. Many of the eubacteria are disease-causing organisms when it comes to other forms of life. In particular, the discriminating feature that allows us to classify living things as eubacteria or archaea is centered around some very simple biochemical comparisons. So the key discriminator is the structure of ribosomal components. As you'll discover later, ribosomes are organelles inside cells which make proteins. By comparing these sequences from different living cells, we can come to a consensus as to how closely related they are simply because of the changes amongst these particular structures. It turns out that all the eubacteria share very closely related sequences when compared to archaea. But amongst the archaea group, they all share sequences with each other more closely related than they are with eubacteria. Another difference is that the gene complement of archaea are completely different in many regards from eubacteria. There's not much overlap in the types of genes that the two domains of life there contain. And one last thing, when archaea are biochemically compared to eubacteria, there are some unique pathways that are not found in eubacteria, which are found in archaea. So these changes in total tell us that the two groups are very different to each other. One thing that the two domains of life do share in common is that their cells come in three basic shapes, but their sizes can vary enormously. So the three shapes are spherical, we call them coccus, i.e. grapes. The second shape is rod-shaped, or bacillus, and the third shape is a spirochete, or a spiral. So if you discover a prokaryote, it will fall into one of these three buckets. This one here is simply another version of the first one. If one was to hypothetically combine all the features found in prokaryotes into a prototypical cell, then this one here would be that representation. It doesn't mean that every cell in prokaryotes has a tail, but those that do, they have very similar tails. So once again, one feature shared by all prokaryotes is that they have no nucleus. There is no nucleus inside this cell. But what they do have is an aggregation of their DNA chromosome into a blob somewhere inside the cytoplasm of the cell. Students mistakenly think that this is a nucleus. It's not. It's simply an area where there's a lot of DNA compared to other parts of that same cell. These cells are typically of the order of 1 to a few micrometers in width or length. They are surrounded, as the definition of living things suggests, by a plasma membrane, here indicated in white. Another feature that most prokaryotic cells have is that they have an extra layer of material outside the plasma membrane, akin to an overcoat, and that is called the cell wall. The cell wall provides additional protection to the cell from the environment. Here we see an E. coli cell. This picture was taken with an electron microscope. The DNA is the light area in the center of the cell, and the area in darkness is the cytoplasm. And these granules that you see are the organelles called ribosomes, which we spoke about earlier. They make protein for this particular cell. The plasma membrane is seen as the white area just on the inside of the cell, and the cell wall is the dark area indicated here. E. coli have no tails, therefore those are not present in this diagram. The scale bar suggests that this cell is approximately 1 micrometer in width and about 3 micrometers in length. Another combination of bacterial cells is indicated here. These are lithotropic bacteria. That means they feed on inorganic material as their source of energy. These particular bacteria would be found in deep oceanic vents under the ocean, and they get their food supply not from sunlight or from carbon, but from sulfur being emitted from the core of the planet. The yellow deposits inside the cells, and this is the cell of the bacteria, there's two cells represented here. The yellow deposits are the sulfur that the cell uses for energy. Finally, this picture here just tells us that there are some extreme cell types on the planet that can withstand very harsh conditions. So this hyperthermophile simply means that these cells are discovered living at extreme temperatures. And in this case, at 121 degrees Celsius, in this example, these cells were found at Yellowstone National Park in some of the geysers. Moving on to talk about eukaryotic cells. Eukaryotic cells, as you now know, share one feature in common. They all contain a nucleus. There are many differences amongst the eukaryotic cells. Some of them have very unusual reproductive strategies. Here we have a mother yeast cell giving rise to a baby yeast cell by a process of budding. So basically, a growth appears on the surface of the mother cell, and that growth increases in size until it pinches off and it releases a baby cell into the environment. And that baby cell will then grow into a mother cell and continue the cycle. This is a higher level exam question where the student will be required to assimilate information from various chapters in order to understand and answer these questions. So the question says, how does cellular specialization serve multicellular organisms, and how might a high degree of specialization be detrimental? So what are the benefits of having multicellular organisms, and what are some of the negatives of having the same multicellular organisms? So this will be an essay type question, and the student is expected to generate an answer of this particular caliber. And you can stop the video here if you want to read the answer. One of the major goals of this course is for students to be able to compare and contrast the eukaryotic with the prokaryotic cells. This table first shows you which features are shared by both prokaryotic and eukaryotic cells. This list is quite comprehensive. It starts with easy to recall information, and then it progresses into more detailed and more difficult concepts. Regardless, students are required to understand this list in its entirety. So these are features which are shared. The second table here is a contrast table. It shows you which features are different between prokaryotic and eukaryotic cells. And once again, we start with a easy to understand concept, and we conclude with some very detailed biochemical comparisons. As we conclude this module, you should now be able to describe which features are shared by all living things. You should also be able to describe which features distinguish living things from each other. Finally, you should be able to describe the number of domains into which modern life is classified, as well as comparing and contrasting eukaryotic and prokaryotic cells. That brings us to the conclusion of Module 2. I think I'll stop the video here and then start a second part for Module 3. Thank you so much.